Okay, thank you very much for the introduction and for giving me the opportunity to present our work here. So in this project, our aim was to expand the therapeutic applicability of bacteriophages that potentially could go way beyond their traditional use as antimicrobials by exploring gene-encoded elements that facilitate interaction with the human body. So what prompted this research is that we know for a long while now that phages are extremely abundant in the human body, but how far-reaching and diverse these interactions can be between the phages and the humans, we only recently started to appreciate. So, for example, while we know that phages in general can influence inflammatory processes, whether they induce anti- or pro-inflammatory responses is highly dependent upon the specific type of phage. We also know that some phages are able to increase the viability of lung epithelial cells by serving as nutrients, while still others have been shown to be able to proficiently cross the otherwise highly selective barriers of the human body, such as the gut epithelium or the blood-brain barrier, so much so that there are no regions in our body that would be left untouched by the phages. Unfortunately, we have very little idea about the identity of such phage-borne drivers, yet knowledge upon these determinants would represent a potential therapeutic goldmine by which we could greatly increase the therapeutic potential of bacteriophages in a variety of manners. So for example, if we could discover and describe the function of such phage-borne elements, we could use them, for example, to measure up how a certain phage intended for therapeutic usage will be able to handle the internal conditions of the human body. Will it be able to cross barriers? Will it be able to hide from the immune system? Will it be able to target intracellular bacteria and so on? And provided that these traits are transferable between phages, we could employ genome engineering to transfer such traits to another phage that lacks some kind of specific feature that we want it to have. And we could even engineer phages by which we could efficiently and precisely carry genetic and/or chemical cargo to hard-to-access areas, be it an organ such as the brain or some kind of intracellular target such as the nucleus or the endoplasmic reticulum. And so the main question of our research was whether we are able to unearth such phage-borne genetic determinants that the phages can universally use to interact with human cells, and whether we can employ genome engineering to transfer these traits between the phages. As a first step, we wanted to identify a specific group of phages that we show are able to interact with epithelial cells, because epithelial layers are known to be one of the most prominent sites of contact between phages and humans. And after that, we wanted to investigate these specific groups of phages for their genetic background to see what makes them special in this regard. So we collected the fecal viral filtrate of different individuals in different states of health, then applied this filtrate on different types of epithelial cell models. After incubation, we separated the interacting or adhering phages from the non-interacting ones, then performed next generation sequencing. And the idea was that we wanted to pinpoint genetic signatures that are characteristic to the epithelial cell interacting phages. To ensure that we are working with high quality sequencing data, we implemented multiple steps of bioinformatic quality control, after which we determined the genetic repertoire of the interacting and non-interacting phages down all the way to the domain level, then compared the results with each other and found that among the adherent or interacting phages, the prevalence of immunoglobulin or Ig-like domains is significantly increased to the compared to the non-interacting ones, implying that one of the prominent conserved function of the Ig-like domains found on the phages is to provide means to for the phages to establish interaction with the human cells, just like how the bacteriophage adherence to mucus model predicted more than a decade ago. Of course, these were just bioinformatic predictions. So next we wanted to experimentally corroborate our results. So we picked K1F phage, K1F model phage that we previously shown to be unable to interact with the epithelial cell layers, and engineered it using in vivo recombinating coupled with CRISPR-Cas9 counter selection to display some of the candidate proteins that we found in the fecal filtrate. This resulted in 3 recombinants, all of which displayed one of the candidate proteins that we found. We also included T4 phage that was previously shown to be able to attach itself to mucosal cell surfaces through its Hock protein that we also engineered to be displayed on the K1F phage. After that, we fluorescently labeled the genome of these phages, applied them to 3 different epithelial cell models, and investigated their behavior under microscopy. So what we found is that all of the recombinant phages show high efficiency to getting internalized by these epithelial cell models, except in the case of the CACO2 that we know has a very low uptake potential. Reassuringly, the positive control T4 and the K1F Hock also gave strong signals, and we also captured this event in a video that I hope I can show you. So in nucleus, you can see the nucleus. In green, you can see the phages. And you can see how the K1F Y-type phage is almost completely unable to internalize the epithelial cells, whereas its exemplar recombinant counterpart can do with a very high efficiency. So we know that these phages are making it into the epithelial cells in one way or another, and we wanted to see what happens with the phages after this. Now the endocytotic pathways can be very complex, and the literature about them is very confusing for me, but to oversimplify things, they all boil down to 3 possible scenarios: degradation, either completely or just partially, non-degradation, or translocation to the other side of the epithelial cell layer. To save time, I'm going to spoil that we were unable to detect any kind of translocating phages that could go through the grown epithelial cell layer, implying that the recombinant phages have some kind of intracellular destination. To investigate this, we fluorescently labeled cell organelles, lysosomes, Golgi apparatus, and the endoplasmic reticulum, and repeated the experiment with the fluorescent labeled cells, phages, and investigated the colocalizational patterns of these signals under microscopy. So in this plot, you can see the degree of the colocalization between phages and the respective cell organelles. And what we found is, as before, the Y-type phages were almost completely unable to internalize into the epithelial cells, but when they did show some kind of signal, it came from within the lysosomes, meaning that even if some K1F phage gallivants into the epithelial cells by just pure chance, they will be most likely broken down. We also observed that, in contrast, the recombinant phages demonstrate some kind of colocalization with the lysosomes, but they mainly can be found in the endoplasmic reticulum. This means that the recombinant phages gain access to degradative, but also most prominently to non-degradative pathways as well, because the routing to endoplasmic reticulum usually happens via non-degradative ways, further implying that these phages might be still intact inside the cells. We also seen some level of colocalization with the Golgi apparatus, but it wasn't too strong, it was just transient. But that doesn't mean that these phages, again, were unable to internalize into the epithelial cells. They could go inside. As you can see in the case of the K1FL recombinant, it's present in the epithelial cells in a very high number. It doesn't colocalize with the signal emanating from the Golgi apparatus, but it beautifully outlines the pattern of the endoplasmic reticulum. Next, we were curious to see whether these recombinants utilize identical or different receptors from each other. So we repeated yet again the experiment with the fluorescent labeled phages, but pre-treated the epithelial models with one of 3 detergent agents that are degrading the epithelial cell surface in a different manner, and wanted to see whether the changes in the internalization rates or dynamics will be different between the different recombinants. Now, this experiment didn't allow us to exactly specify the identity of the receptor on the human cell surface, but it did show us that since all of the recombinants reacted differently due to these treatments, that most probably these recombinants all use different receptors for internalization on the human cell surfaces. And last, we also wanted to see whether our modification have had any in vivo effect. So we fed mice with wild type or one of the recombinant phages. And after, at set time points, we collected the feces, measured their phage content, and based on that, investigated how quickly or slowly the phages are flushed out from the mouse system. We found that, as expected, the K1F phages quickly, relatively quickly, in just a few hours, appeared in the stool of the mouse, as written in the literature. However, in the case of the recombinant phages, they almost never showed up, or just in a very minimal amount, implying that due to the modifications, either they managed to interact with the epithelial cell layers of the mouse, or some other reason, but because of the modification, just stuck inside the mice. And the direct follow-up of this work, the study, will be to chemically conjugate therapeutic drugs on these endoplasmic reticulum internalizing, localizing phages, and use them to try and treat disorders that in which the endoplasmic reticulum is involved in, such as cystic fibrosis. And also parallel with this project, we also working on phages to, with the very same approach, to get through phages the blood-brain barrier. If for details, if you haven't done so already, I heartily recommend to visit the poster of the Dr. Dóra Sala. And with that, I would like to thank all my friends, colleagues, and collaborators, and thank you for your kind attention.